Hello everyone. So let me just check if I'm live. Okay, so there is error. All right. I think now it is fine. Okay, so before I start, let me introduce myself. I'm Vani Kari, biology educator at Unacademy. I'm an engineer with bachelor's and master's degree in biotechnology and a B.Ed degree. Also a founder of a startup where I grow microgreens by vertical farming technology. So just a second, I'll just check this. All right. Okay, so join with us in Telegram for homework and class notes. You can join the channel Vani Kari class 9 and 10 biology and the Telegram channel of Unacademy, ICSE and ICSE both. So this link you can find in the description box. Okay, so go to the description box, find the link there, and basically you will be getting the updates regarding the classes that are done here on the YouTube. Okay, also we conduct special classes. Just a second. Yeah. Yep. So also we conduct special classes on Unacademy platform which are absolutely free. So you can learn from your favorite educator for free, engage with educators via poll. That means basically we'll be conducting a quiz. So once we conduct the quiz, so at the end of the quiz we'll be sharing a leaderboard so that you can know your rank and compete with your friends. Hi, hello, good evening everyone. So clear your doubts in the live special class. So there is a question tab where you have an option to attach an image, and you can also type in your question and send it to the educator during a live session. So they'll be getting a notification that the learner has asked the doubt, and they'll be clearing your doubts during the live session. Okay, now for the top special classes, again you can visit the link that you can find in the description box. So Unacademy subscription feature, which are about the plus classes, here these are free classes. Okay, so the special classes are free classes. So here in this plus class, a complete syllabus will be done. So main thing is you can learn live from the comfort of your home. Unlimited access to all the courses will be there. So here different educators will come up with different courses. So you will have access to all the courses run by the educators once you take the subscription. Then top educators of India are going to take the classes in one platform, that is Unacademy platform. There is regular doubt clearing session. So that is after about every 3 sessions. The fourth session will be a doubt clearing session. Okay, so whatever doubts you have related to the topics that are previously covered in this 3 session, so you can clear your doubt in this session. So that is a doubt clearing session. So if no doubts, basically we can discuss the Selena solutions as well, and along with that, so we'll be conducting a quiz also. Okay, so this is a totally a discussion session which is meant for learners to clear their doubts. Then complete syllabus will be covered. Here in special classes only a certain percentage is covered. So random topics are chosen and the classes are taken. But the plus classes, so they'll have a fixed timing for each and every subject, and a whole syllabus 100% syllabus coverage will be there in a plus class. Mentorship and guidance is given, study material is given, so that is in the form of a PDF. Then practice tests are given, live test series are given, and along with that, so there are batches as well. So there is a batch in English, so there is a batch in English. So once you take the subscription, so you will have access to English batch also, Now depending on your comfort level, so you can go with any of these batches. Then there is daily practice section, weekly mock test series, and with that there is live quizzes, daily MCQ and subjective test series are also conducted. This is, since this is the June month, very beginning of your academic year, so you can go with 12 months of subscription, or you can also even try with 9 months of subscription. So if you just want to check like how exactly this works, so you can try with 6 months as well. So now for 12 months, so basically it is 17,250, but with the referral code Vani YT, so you will be getting a 10% discount on the subscription. So 10% off you are getting on this amount, that is 17k, so that is 17,250. So those who are planning for 12 months, so basically along with 12 months, so you will be getting subscription for class 10, class 11 and class 12 as well. So whenever you have time, so in the meanwhile you can prepare for those sessions, as those classes as well. So you can even attend plus classes where the 100% syllabus coverage will be done. So those who are planning to go for the subscription, so don't forget to mention the code Vani YT here in the gift box, that is add a referral code. So there you need to mention where you will be getting a discount on your subscription. Alright. So today we are going to discuss about the structure of a cell. Just a second. Alright. So, alright. Okay, so structure of a cell, when we mention, what all do you find in a cell? Anybody? So what are the three main components that we come across in a cell? So we'll be discussing about the structure and function, but mainly when we mention there is a cell, so what exactly is present in a cell? So there is a cell membrane, okay? So this is common in all the living organisms. So basically there is a cell membrane will be there, okay? Then along with cell membranes you can also find there is a nucleus that is present, and here there is a semi-liquid that is present. So there is a semi-liquid, so that we call as a plasma, cytoplasm, okay? So it is basically a cytoplasm which is present here inside completely. So this whole space what you see, so this is completely filled with cytoplasm, okay? So that means there is a cell. So cell consists of three main basic components that are there are the cell membrane, the cytoplasm, the nucleus you can find, and along with nucleus, so basically the genetic material that can be seen here in the cell, okay? So now this can be the cell can be of any shape. So you have come across many different types of cells. So mainly there is a irregular shape cell like amoeboid cell, basically WBC we can say, or maybe a amoeba itself or a slime mold. So these are all amoeboid. Then there is RBC, so which is a dish shaped one, okay? So like depending on the structure and depending on a function, so basically there are a lot of changes that we can see. So now if a particular function has to be done by a cell, so there is a structure, so that structure is going to help in that particular function. So depending on that there is a size variation, structural variation, and today's session will be mainly discussing about the cell. So cell being having its different structure. So let me just write this. So we'll see like the cell structure what is there, and the organelles, basically the structure and function when we mention. So this is with respect to the organelles, okay? So organelles, their structure and functions we'll discuss in today's session. All right, so this is mainly a cell membrane, okay, or which can even be called as a plasma membrane. So this part is what? This one is a nucleus, and here this semi-fluid what is there? So that is a cytoplasm. So this is what is written here in the previous slide. So main features of a cell when we mention, so there is a cell membrane, there is a nucleus and a cytoplasm. Then what is present here inside this? So that is inside the cytoplasm, what do you see? So in the cytoplasm, yes, anybody, what do we see in the cytoplasm? Any of you? No idea? Okay, so basically in the cytoplasm, hi, good evening. Yes. So in cytoplasm there are many different cell organelles that are there. Okay. So these cell organelles are the ones that we are going to discuss today. So that is about the structure and their function. So how exactly they are arranged. So now specifically with respect to eukaryotic cells we'll see. So before that, let's see what exactly is a cell organelle. So cell is composed of several membrane bound structures. So what are those membrane bound structures? So let's say here one membrane bound structure is there. So there is a outer membrane and the inner membrane is actually folded randomly. So this is what? So this one is actually a mitochondria. Okay. So now you know outer membrane is there, inner membrane. So that is again a membrane bound structure itself. So cell organelles when we mention, so several membrane membrane bound structures are found and these structures are going to perform the specific function. Okay. So that specific function here with respect to mitochondria when we mention, so there are a lot of mainly the respiration, the energy synthesis what we mentioned, so enzyme synthesis in certain cases, DNA, it carries its own genetic material. So all that could be the reason for this one. Okay. So now organelles, what are those organelles? So basically nucleus, a plasma membrane, a mitochondria is present, Golgi body is present, and then there is a vacuole. Okay. So all these you will come across. So that is in case of organelles found here in the cell. So let's see this. So that is about the eukaryotic cell. So what is eukaryotic cell? The size, the nucleus, how exactly it is present, or why do we call this as a eukaryote? Any idea why do we call this as a eu? And what is the meaning of karyote or a karyotic? Any of you? No idea? Okay, so I'll just mention. So U when we mention, so U is basically true. Okay, so there is a true nucleus. So U is true. The karyos is basically a nucleus. So true nucleus. Why do we say it is a true nucleus? Okay, prokaryote, yes, that is prokaryote, we call a primitive type of nucleus. So that is this type of nucleus you can find in the older organisms as well. So that is the bacteria. Okay, so in that case that is a primitive nucleus. Here this one is a true nucleus, which means it is a well defined nucleus. Okay, so this one is a well defined nucleus, whereas the other one, which is that other one mainly. So now when we mention about the prokaryotic, so that is the primitive nucleus, or basically which you can find in bacterias, mainly all the moneras we can say. Okay, so they are all having a prokaryotic arrangement, rest of them are all eukaryotic where their nucleus is well defined. So what is the meaning of that well defined nucleus? So let's see this. I'll just draw here and explain you like what is the structure. So now this is the cell membrane. Okay, so cell membrane is found, and now what we are discussing, so we are saying, we are talking about the eukaryotes. So why we are saying eukaryote? Because it is having a well defined nucleus. So what exactly is that well defined nucleus? I'll just draw it and show you like what is well defined nucleus here. Okay, so now there is a nuclear membrane. So there is a outer membrane, like this. So this one is the outer membrane, and then you can find the inner membrane. Alright, so there is the inner membrane as well. So now this one is the inner membrane here. So outer membrane and inner membrane is present, and then why I am drawing a gap here? So any idea why I am showing a gap over here? Hi Aryan. So this is, the outer membrane and the inner membrane have fused. So now when you see a structure of a well-defined nucleus, so basically you can find the nuclear envelope is having a outer membrane and the inner membrane, and this gap what you see is basically a nuclear pore. Okay, so this is what? This is basically a nuclear pore. All right, so there is a nuclear pore, there is a outer membrane, there is a inner membrane, and inside the structure itself you can find a chromatin fiber or DNA that is suspended. Okay, so now this is the DNA that you can find. So that means, so DNA is there and it is not freely suspended in the cytoplasm. So there is a covering, and that is the reason we call them as a eukaryotic organism where their DNA is well defined one. Okay, so there is a covering, and now inside this structure again you can find one more thing is basically a nucleolus. Any idea what does the nucleolus does? Yes, that's nuclear pore. So this one is nuclear pore. Okay, so now this one, the black one what I have labeled here, so this one is a nucleolus. So any idea what does the nucleolus does? So this is a well-defined nucleus. So why are we saying it is a well-defined nucleus? Because this genetic material what you find here, so this is covered by a membrane. So there is a membrane, and that's the reason we call it as a well-defined nucleus. Then what about in case of bacteria? So now bacteria, when we mention, DNA is actually suspended freely here. Randomly DNA is present. Okay, and there is no covering for this one. So this is a primitive type. So primitive type of bacteria or primitive type of organisms when we mention, so their genetic material or a DNA is randomly suspended here in the cytoplasm itself. But here in this case what is happening? The genetic material, the DNA or a chromatin fiber what we call, so that is completely covered by a membrane. So that we call as a well defined nucleus. So let's see some features that is related with the nucleus. One second. What is the full form of DNA? So it is deoxyribonucleic acid. So Muhammad has answered. That's right. So nucleolus helps in protein synthesis. Nucleolus is going to help in the synthesis of ribosomes. Okay, so this function is ribosome synthesis. Okay, so when the ribosomes, as you all might be having an idea, ribosome, just who said that? Aryan. Yeah, so Aryan just said that the ribosome helps, basically nucleolus helps in protein synthesis. It is partially true. Why? Because nucleolus is synthesizing a ribosome, and ribosome in turn is synthesizing a protein. Okay, so in that case what will happen, as the protein is being synthesized by the ribosome, but the ribosome is in turn synthesized by nucleolus, so they are again one and the other are related. So let's say I'll just show you this picture now. One second. So now this is the nucleolus. So let's say ribosomes are synthesized by this one. So these ribosomes, that you always label the ribosomes here in the cytoplasm, right? So why are we showing a ribosome in the cytoplasm even though it is synthesized here in the nucleolus? So that is because these ribosomes which are synthesized, so they will move out through the nuclear pore. Okay, and they are freely suspended throughout the cytoplasm. So now this part what is done is, So this one is the ribosome, okay, and main function of ribosome is what? It is a protein synthesis. So protein synthesis is done by the ribosome. Now in continuation, so there are different organelles, we'll discuss about that. Yes. So basically, yes, and also contains RNA. Yes, basically, the ribosome is made of a RNA itself. So there is RNA, and this RNA. So if you know this, so basically we call that process as a central dogma. So what is the central dogma is? You know, DNA is there in your body, that is in each and every cell. So this DNA is coding, okay, so this is coding for the RNA synthesis, and RNA is coding for the protein synthesis. So that means what? So this ribosome that we are talking about, so ribosome is basically a RNA. It is made of RNA itself, and that RNA is coding for protein. So DNA is coding for RNA, and RNA is coding for the protein synthesis. So like, you know, each one of these are going to help. So how exactly this will happen? So this will happen with the help of the enzymes that are there in a each and every cell. So now these are the ribosomes which have a role with respect to protein synthesis. So let's have a look at the endoplasmic reticulum. Okay. Yes. Alright. So now here in this case, so let's see this. There is a next structure. So there is a next organelle. So which is the endoplasmic reticulum, which lies, you know, which starts continuously from the outer membrane. So we can just show the endoplasmic reticulum like this. Okay. So which is in continuation from the outer membrane itself. So that is the outer membrane of the nucleus. Okay, so then, you know, you can see this is the endoplasmic reticulum. But in certain cases you might have come across the term like there is a rough endoplasmic reticulum. Any idea why do we call it as a rough endoplasmic reticulum? Yes, there are two types of endoplasmic reticulum. So one is smooth endoplasmic reticulum and the rough endoplasmic reticulum. That's right. Okay. So in that case, now any idea? So one more question. So any idea why exactly do we call this as an endoplasmic reticulum? So do you understand? You can just go ahead with the name. Okay. So endo is mentioned, plasmic is mentioned, and reticulum. So anything that comes to your mind. So along with, like, you know, if you go word to word meaning. So what is the meaning of endoplasmic reticulum? Because it is having a ribosome. Yes, so that is a rough endoplasmic reticulum, because when we observe it under the microscope, you know, this gives a rough appearance. So this part, what I am showing a ribosomes here, so this will give us a rough appearance. But if we observe on this side, okay, so it can be, you know, it need not be here always. Here also ribosomes might be attached. So some parts the ribosomes are not attached. So now, you know, this part what you see, so this is actually the smooth endoplasmic reticulum, because there are no ribosome. And here this part what you see, so this one, or maybe I can just show it here itself. So this is a rough endoplasmic reticulum, because it contains ribosomes on the surface. Okay, so why exactly name endoplasmic reticulum? I'll just say, okay, so endo is what? Inside. Plasmic is what? Inside the cytoplasm. So now you can see this structure inside the cytoplasm. And what is reticulum? So reticulum is basically a network like structure. So this network like structure you can see inside the cytoplasm, and hence the name is given as endoplasmic reticulum. Okay, so now what exactly is the function of endoplasmic reticulum? That's nice, Mohammed. That's nice. So you have just come to revise it. Okay. So any of you, or like, you know, get any doubt, you can just ask me here itself. So I'll just clear it out. Okay. So now here, you know, This is the endoplasmic reticulum. The function of endoplasmic reticulum is that it is providing a supportive framework to the cell. Okay, so since it is forming a network area, here I have drawn only one. So you can see the endoplasmic reticulum, which can continue from here also, it can continue from here, then it can extend till the cell membrane as well. Okay, so that means it is a continuous framework and it is providing a supportive framework for the cell. So that is one of the function that we can say with respect to endoplasmic reticulum. So then transport of material throughout the cell. Yes, that's right. Okay, so I'll just tell you how. So it is not exactly modification we can say. So transportation is also done. So now let's say here I'll just show you this. So now here the protein is synthesized. So that is inside the nucleus. As you can see, already a ribosome is present there. So this ribosome which is present, so that will produce a protein. So now this protein which is produced, so this is going to take an entry over here, or maybe from the ribosome here the protein is can take an entry. And here in the endoplasmic reticulum, so this is about endoplasmic reticulum, I'll just write it here. Yeah, so here in the endoplasmic reticulum what exactly happens is modification of protein you can see. So transportation is also occurring, so modification of proteins and lipids can be seen. So what is the meaning of modification? Any idea? So protein and lipid modification is done here in the endoplasmic reticulum. Yes, so transportation of the material throughout the cell. So why? Because it is a network like structure. Reticulum is what? It is a network like structure. So once the things are synthesized, so these things can move in and around. So basically these are transported across this particular endoplasmic reticulum itself. So here I was saying the modification of protein and lipid. Any idea what is modification of protein or lipid? What is done to that? No idea? Okay, I'll just explain you this. So what exactly is modification of protein is? Now let's say protein is synthesized. So for that protein we want to add some carbohydrates. Okay, so some carbohydrate has to be added. So let's say this is a protein chain that is present. Okay, so this is a protein chain. If we add a carbohydrate chain here, okay, or if we add a not glucose, mainly we can see the lipids. Okay, so lipid chain is added here. So in that case, basically depending on what is added, so that here let's say lipid is added to the protein, so that we call as a lipoprotein. Okay, so this one is a lipoprotein. Here the carbohydrate is added to this protein, and that is why we call this as a glycoprotein. Okay, so this way modification what occurs with respect to the what we see. So this is protein which is synthesized, so that is getting modified, and the transportation is done to the next organelle, that is in continuation. So which is that next organelle? So in continuation with this organelle, so you can always find a Golgi body. So there is a Golgi body here. So you are in 8th standard, finished class 8 syllabus and studying class 9 and 10 syllabus. Okay. So you finished class 8 just now. Okay. So since this is the very beginning, yeah. So basically you can start ahead. So genetics class 10 ICSE. Genetics mind map shall I discuss in the next session? So not tomorrow's session is anyway decided. So tomorrow is what day? So tomorrow is Wednesday, right? So maybe Wednesday, Thursday I'll plan. Okay, so Thursday I'll just discuss the mind map of the genetics. So you can ask whatever doubts you have there. Okay. So you are you are studying for UPSC. That's nice. So you all have a good goal. Okay. So now as the protein gets modified here, or as the lipids get modified here, so this modified protein which is this, so that is going to take an entry here into the Golgi body. So Golgi body function is what? So this is a Golgi apparatus. Okay. So this Golgi apparatus, when we mention, so it is having a vesicles as such. So there are vesicles. So these vesicles, when we mention, so now the protein which was modified, so it is going to take an entry here, and the again further modification and packaging is the main function. So like it is given in your textbook, what is mentioned, the Golgi apparatus function is the acrosome synthesis. So enzymes in the acrosome. Okay, so that is the acrosome of a sperm basically. So this is a acrosome of sperm, contains the hydrolytic enzymes. Okay, so what is enzyme in nature? So if you have studied, so enzymes are basically protein in nature. So what is done in the endoplasmic reticulum, that is the protein modification was done. So further protein modification will be done, and that will be made into a enzyme there. Okay, so that is a hydrolytic enzyme which is beneficial for the penetration of the ovum. So that is across across many of the sperms that are competing. So one of the sperm is going to unite with the egg. So in that case, the acrosome of a sperm will have a hydrolytic enzymes, and these are going to break through the wall of the ovum, and then the fertilization can be seen. Okay, so that's the reason. So acrosome is one such function. So as such, many other organelles that are there. So let's say lysosome formation is required. Seed germination, class 9. So you want me to go ahead with seed germination? Okay, so I'll plan it. Okay, so Thursday I told genetic mind map. No, so if there is one more class that I can take on Thursday, so maybe germination I can go ahead with. All right. Okay, so now here in this case, just a second. All right, so now as the proteins which are modified, so they are reaching this part. So there is further modification and the packaging is done. So this one is a Golgi body now. So in the Golgi body you can see a packaging of the material, like whatever modified protein. So this can be enzyme, this can be hormone, so this can even be a lysosome. Okay, so lysosome is basically what it is, a suicide bag, right? So now Golgi apparatus itself is packing all the different enzymes in the form of a vesicle, and now you can see basically arising from this structure. So you can see there is a lysosome formation. So let's say here this one is basically a lysosome formation that you can see, which is called as a suicide bag. So why do we call it as a suicide bag? So whenever there is a germ entering our body, okay, or if there is a old organelle, let's say mitochondria might be old, endoplasmic reticulum might be old. So whenever old organelles are present, so in that case also this lysosome is going to digest it, it is going to kill it. Okay, and other than that, the main function of this particular lysosome, when we mention, so I'll just label this one. So this one is lysosome. Okay, so hydrolytic function is done. So this is basically whenever the unwanted things like foreign materials or allergic reaction, this won't have a major role as such, but yes, this is having a role with respect to allergic reaction and the pathogen entry, old organelles that are there, damaged cell is there. So now this particular lysosome is going to release the enzyme to the surrounding, so that is by bursting, and rest of the things are gonna digest it. Okay, so cellular digestion is the main function, so that is associated with the lysosome. And the Golgi apparatus can also give rise to a vesicle. So let me just change the color for vesicle. Just a second. 1 second. What is this? Why is it blocking? Okay, so now this part what you see, so this is basically a vacuole formation might be there. Okay, so now in this case, just a second. So am I audible? So I think some network error is there because I'm getting a side sound from the audio here. So am I audible clearly? Bullet Gamer or Mohammed? Yes, okay, thank you, Gamer. Okay, so here in this case, the Golgi apparatus itself is giving rise to a vacuole. So now, in certain cases, what will happen? The Golgi body, let's say this is an animal cell, since we have not drawn any of the cell wall. So you can find smaller vacuoles can be there. Okay, so smaller vacuole can be produced from this Golgi apparatus, or it is completely absent also. So if a vacuole is present, so vacuole is again covered by a membrane. So there is a membrane bound structure, and here you can see some stored things. Okay, like required stored things, like so there might be requirement of water or some food granule storage. So vesicles we can call them. So this can even be a vesicle that is mentioned. Lysosome is going to commit suicide. So that is because all the unwanted things have entered the cell. So this is going to burst open. The lysosome is going to burst open, and the cellular digestion. So function of this one is cellular content digestion. So whenever there is basically a pathogen attack, we can say. So now these are the organelles, and other than that, mainly you can see a mitochondria in a cell. So I'll just draw a mitochondria here. So this one is a mitochondria, which is having the outer membrane and is having the inner membrane. So this one is the outer membrane. Now inner membrane has folded, like it has a folding, and these foldings are called as a cristae. Okay, so now this one is outer membrane, and here what you see, so this is the inner membrane. Now, when we mention that the mitochondria is having its own DNA, so just like the nucleus having its own DNA, mitochondria is also having its own DNA here in the matrix. So now this whole part is basically, so I'll just highlight it just a second. Okay, so now you see this part. So this part is what? There is a outer membrane and inner membrane. So there is this space is there, no? So the space is called as a intercellular space here in the mitochondria alone. Now inside this space, I'll just change the color. So this space what you see, so that is basically a matrix. Okay, so that is called as a matrix. So here this I'll just change the color. Which one shall I take? Okay, so now this inside part what you see, so this is basically, you can see this as a matrix, and inside this matrix itself, so you will come across a genetic material, so that is a DNA. Okay, so here you can find, one second, so it is not even clearly seen. Yeah, so now here you can find basically the ribosomes that are freely suspended, and other than that, so there is a DNA that is present. So mitochondria is an independent organelle where it is having its own DNA, and it is also having its own ribosome. So it can undergo protein synthesis. So that is DNA is coding for a RNA, and RNA is coding for a protein. So let's draw the DNA here itself. So some randomly arranged structures, so that is the DNA is present. So now what exactly is the function of this mitochondria? Any idea? What is the function of mitochondria? Yeah, so this one you want me to label. So this part is there, no? So this foldings what are there? So this inner space is basically a matrix, and these foldings what you see, so these are actually the cristae. Okay, so I'll just label one here itself. Just a second. So this is actually a cristae. So this part what you see, so these foldings in the membrane what are there? So those are cristae. Okay, and inside that the matrix is present. So that matrix is the one which is having a suspended DNA and along with the ribosome. Now this whole structure is a mitochondria. The main function is basically, yes, that's right. So that is a powerhouse. So why do we call this as the powerhouse of the cell. So why this is producing so much of energy? So we'll just see that. I'll just explain it to you here. So mitochondria is called as a powerhouse of the cell. Okay. So now why exactly do we call this as a powerhouse of the cell? Because this is getting oxidized by the glucose. Okay. So that is here, glucose oxidation you can see in the mitochondria itself, which is going to produce a carbon dioxide. So just like we respire, so we are taking a oxygen and leaving out a carbon dioxide, right? So same way here in the mitochondria, when the cellular respiration occurs, so this is with respect to cellular respiration. Okay, so here what will happen, oxidation of a glucose can be seen. So there is a carbon dioxide, there is a water, and then there is a ATP that is produced. Okay, so that is the adenosine triphosphate, and this ATP is the energy currency. So now to perform any activities, now we are all performing a certain activity. Let's say you are playing in a ground. Okay, so you are running, maybe you are playing a cricket, maybe you are playing a football. So any of the activity you require energy. So if you haven't had food, so there is no energy. So now once you take that food, so that will be broken down into glucose, and that glucose is gonna join with the oxygen. That is oxidation of a glucose can be seen, and these are all the waste products which are removed out. So if you just hold your hand to the mouth and you exhale, okay, so that you are respiring out, so basically you can feel that the air is there and some moisture also. Or if on glasses, if you just blow up the air, okay, so you can see after some time, so basically you can see some dot or vapor like structure. So all the water, I mean vapor, has condensed to form that structure. Okay, so in that case the oxidation of glucose will be forming the carbon dioxide, which is a waste product. Okay, so this one is a waste product. So you can, when you respire, so water is also moving out through the respiration to some extent, not whole water. Then there is a ATP, energy currency. So now as you get the energy for any activities, basically, so your muscles can move. So there is a muscle movement, so there is a thinking capacity, logical reasoning. So for everything energy is required, and with that source of energy which is provided by the mitochondria, and so this will be used in many more activities. So with respect to that, we can say mitochondria is basically a powerhouse of the cell. Can you please explain the endosymbiotic theory of mitochondria? So this is in class 12, no? Aryan? So Aryan has started studying class 12 also already. So I'll explain. So now earlier this cell organelle, that is mitochondria, was living freely. Okay, just like this. So I'll just explain you this. Okay, so now let's say there is a mitochondria. Okay, so this mitochondria having a inner membrane, so that is folded as such. So this one is a mitochondria. So this was free living. So as you can see already, it is having its own ribosomes are there. So then it is also having its own DNA as well. So in that case, earlier this was a freely living structure. So that is a mitochondria. So now as you can see, there is a bacteria. So bacteria engulfed this one. So that is bacteria took this one, and that is how endosymbiotic theory says what is the prokaryotic cell itself has has evolutionized, and it has formed the eukaryotic cell. So that is by engulfing or maybe taking in some of the organelles, where they started to develop a mitochondria, where they started to develop the endoplasmic reticulum, where they started to develop the different cell organelles. So that is because prokaryotic cell, the bacteria itself, is basically engulfing this, and there is a formation of the organelle. So now both are getting benefited mutually, and hence the reason. So now we have this eukaryotic cells. Okay, so now components of eukaryotic cell when we mention, I hope it is clear for Aryan. Just a second. Okay, so now what all components you can find in a eukaryotic cell? So there is a cell wall, there is a plastid, okay, then there is a cell membrane, there you can find a ribosome. So plastid, basically you can find a chloroplast, okay, so which is having a green pigment chlorophyll, okay, so this one is having a chlorophyll, a green pigment for the photosynthesis. Then there is one more plastid, so that is a leucoplast, which is not having any type of pigment for photosynthesis. So what it does is the food storage can be done, okay, so this does the function of food storage as well. Then there is a chromoplast. So what exactly is chromoplast? So any colored body, so let's say there is a carotenoid pigment, okay, so carotenoid pigment might be there, or we can say anthocyanin pigment, the xanthophyll pigment, so this can be anthocyanin, then there can be xanthophyll, okay. So whenever we are mentioning about the plastids, so you can find a different organelles there. So there is a chromoplast, there is a chloroplast and a leucoplast, which is storing the food, storage is done, and here green pigment photosynthesis is done, and this one is a carotenoid, so or we can also call it as a theory gain concept. Then one second, just a second. And then you can also find there is a anthocyanin pigment, so xanthophyll pigment, so different pigments. So now orangish color, yellow coloration, the purple green coloration, so somewhat bluish coloration, so depending on what type of a pigment is there, so these chromoplasts are going to store this different pigments, okay. So then endomembrane system. So what is endomembrane system? Under endomembrane system we just saw there is a endoplasmic reticulum, the Golgi body, then the lysosome also comes, the vacuole also comes, okay. So now plant cell vacuole when we mention, so in case of plant cell vacuole, so let's say this is a plant cell, so there is a cell wall, so there is a cell membrane that you can find, so this one is a cell membrane, and 90% of the space is covered in case of a plant cell, so that is by a vacuole itself. So what exactly does the vacuole contain then? Vacuole is having water, okay, so some water, so some salts might be there, and along with that some dissolved pigments like anthocyanin are also present here. So this is a anthocyanin, okay, so this pigment can also be present here in the vacuole. So this is a larger, covering about 90% of the total volume or total space of the plant cell is covered by a vacuole alone. Then in our case, it can be completely absent in case of animals, or tiny vacuoles can be seen. So then there is cytoskeleton. So what is that cytoskeleton? So basically when the cell division occurs, so you can see a formation of spindle fiber and all, okay, which will try to, so if you haven't studied, so that is fine. So basically there is a thread like structure formation which will separate the two cells, so that is from each cell two daughter cells are being formed there. Yes, so ma'am, how can we get recorded classes for 9 on Unacademy? So what you can do is the recorded sessions will be there. So if you are asking about free sessions, all you need to do is download the Unacademy Learner app and start attending the session. So once you download the app, so basically this all the regular apps whatever they ask, like get started, you can enter your mobile number, all that you will do, okay. Then you can, now if you are looking for let's say bio classes, so that is mainly biology, some of the recorded sessions that are there, so these are for free sessions. So in that case you can just find my name, you can just search Vani Kari, my name, okay, and there you can also, once you find, so there in under this section, so you can find a special class section. Okay, so the special class section, so whatever recordings are there, so that you can attend. So if you are asking about the plus classes for 100% syllabus coverage, so in that case you can go ahead with the subscription. So you can use the code Vani YT. So this is my code. Okay, so there is no gap. So with this code, basically you can take the subscription where you will be getting a 10% discount on the subscription. Alright, so now here, this is with respect to the cytoskeleton. So whenever cell has to divide, so this is going to form a spindle fiber and a division of a cell can be seen. So nucleus is basically a boss of a cell. So this is the control center. So why do we say that this is the control center? Because the genetic material is present here in the nucleus, and that genetic material is actually making the cell, code for the RNA, then code for the protein. So with respect to all that, what will happen? So there is a protein synthesis that keeps on. So now whatever body features, so now like, let's say with respect to hair only. So some are having a straight hair, so some are having a curly hair, so some might be having a wavy hair. So that is completely depending on a protein that is expressed in their body. So external feature of an individual is gonna vary. So now with respect to that, so now so some will have a brown eye again, so some will have a black eye. So this is again depending on what type of protein is expressed. So whatever body feature that we are getting, so in that case the nucleus is there, and in that nucleus you can find the DNA, and DNA is the one which is maintaining the regular activities that are occurring in a cell. So now mitochondria you can see, which we already discussed, that this is the powerhouse of the cell. I hope this is clear. Okay. So now this is called the powerhouse because this produces the energy ATP. Alright, just a second. Okay. Now cilia and flagella. So what exactly is cilia or a flagella? So let's say there is a paramecium maybe. So let's say there is a paramecium that is present. Okay. So now this paramecium, when we mention, so it is having a cilia growth throughout its body. So which is going to help with respect to, here is this one. So you can find a contractile vacuole here. Okay, so now this tiny hair like structure that you find on this structure. So this one is a paramecium. So this one I have just drawn it like a euglena. So maybe we can just change it a little bit. Look, just we can just show it like this. So this will become the paramecium. Okay, so you can just show it like this, and throughout this one is covered by this one. Okay, so that is the cilia, which is going to help in the movement. Now euglena, if we have to draw, so euglena is like this, having a structure as such, and then there is a flagella there. Okay, so flagella is there for movement, and this is a nucleus. Here also nucleus will be there, but the contractile vacuole is also present. So this is completely a cytoplasm. So as usual, here also you can find it. So now the flagella or the cilia are going to help in the movement of the particular organism. So I hope it is clear. Any doubt? So now a protoplast is a cell. Okay, so this is a protoplast is a cell. So what exactly is a protoplasm? So protoplasm, I already told you, like, now the cell membrane is there. So in the previous sessions also I have explained you this one. So that is cell membrane is present. So inside this cell membrane, what do you find? So there is a cytoplasm, and then there is a nucleus. So that is basically a protoplasm. So that is without the nucleus undergoing division and without the cell wall. So this one is protoplast is without the cell wall. So cell wall is not there. The cell membrane, yes Aryan, that's right. Okay, so in that case the cell membrane is present, the cytoplasm is present, and you can see a nucleus. So this whole thing can be basically an animal cell itself, which can directly be called as a protoplasm. Okay, or a protoplast as well. So controlling center of the cell, Nucleus, nucleolus, mitochondria and ribosome. So which option is correct here for the controlling center of cell? A. Yes, that's right. Okay. So nucleus is the controlling center of the cell. The nucleoplasm is continuous. Okay, so nucleoplasm which is present in the nucleus, so that is continuous with the cytoplasms of a cell, so that is through centriole, endoplasmic reticulum, nuclear pore and the Golgi apparatus. So which is the answer for this one? No idea? Okay, so this is basically a nuclear pore. So here I had just drawn. So now you know in this one, so there is a nuclear pore. So this is what? These are all nuclear pores. So nucleoplasm is in continuation. So that is with the cytoplasm. So that can happen through the nuclear pore. Okay, so nuclear pore is the one which is maintaining that continuity here in the a cell. So the main organelle involved in modification and routing of newly synthesized proteins to their destination is chloroplast, mitochondria, lysosome and endoplasmic reticulum. So which is that? Which is doing a modification and routing of newly synthesized protein. So there are options. Yes, so this is D. Okay, so mitochondria is a powerhouse. What I said is in the endoplasmic reticulum, the modification of protein will be done, which is later on transported to the Golgi apparatus and the packing will be done. So in that case, newly synthesized proteins are modified here in the endoplasmic reticulum. So this is the right answer. So Golgi body originated from lysosome, endoplasmic reticulum, mitochondria and cell membrane. Any idea what is the answer for this? So this is basically a endoplasmic reticulum itself. Yes, that's right again. Yes. So Golgi bodies originated from the endoplasmic reticulum where the packaging will be done. So this is the right one. Okay, so we are done with today's session. As the time is up, so I'll have to end this session. And at 5:15, no, not 5:15, 6:30, I have one more session where I'll be discussing on mind map of cell division, cell structure chapter. Okay, so before I end this, so there is a guardians batch. The mode of delivery is English here. So PCMB are covered in this batch, and if you are subscribing for class 9, you will be getting class 10 to 12 subscription absolutely free, and you can use my code Vani YT. So this is my code. So with this code you will be getting 10% discount on the subscription. Alright, so introducing community. So never miss on any update from your favorite educator. Get session notes and practice material. Then share your feedback directly with the educator via poll. So the link you can find in the description again. So this is opportunity for all learners to report any inappropriate content in the video. So be the first one to report a particular issue to claim your prize. Report any inappropriate content using the form in the description. So see you all. Yes, you have done all the questions correct, Aryan. Yes, that's right. Okay. All right then. So see you all in the next session. So next session at 6:30 p.m. today itself. So that is on cell cycle. So mainly the mind mapping I'll be discussing about. Okay. So subscribe to our channel and click on bell icon to get notified about the classes. Like and share the video and download the Unacademy Learner app. So see you all in the next session. Bye bye everyone. Thank you so much.